Hello everyone, and I hope that you're doing well. Now today we're doing another CSEC past paper review for biology, and we're doing a paper 2 from the year 2017. Now there are a few things I want to explain by going through this paper, but before we start, I want you to remember that when you're going to do the exam, you should read through the instructions carefully and ensure that you understand each instruction. And if you should have any questions or queries, then you clarify those with the examiner before the examination begins. Now let's jump into section A, question number 1. And question number 1, part A, reads that in an experiment, some student put corn seeds in each of the two test tubes labeled A and B as shown in figure 1. I want you to observe the diagram and notice that test tube A has 20 degrees, test tube B 10 degrees Celsius. We have moist cotton wool in both of them, and of course the corn seed will be placed in each of the test tube as well. Now, part 1 is a write a suitable aim for this experiment. And again, if you notice the diagram as mentioned, that the temperature is the only thing that is different. So a suitable aim could have been to determine the effect of temperature on the germination of corn seeds. And for part 2 is in which test tubes A or B would germination most likely occur. Give one reason for your answer. And so I choose test tube A, and again, test tube A, let's go back 1 second. And test tube A, as you notice here, has the highest temperature, which is 20 degrees Celsius. Not too hot, not too cold. But 10 degrees Celsius is pretty much a little bit chill, all right? And so, here now, choosing test tube A, the reason for that is simply because the enzymes would be more active at 20 degrees compared to 10 degrees, where they would be less active. And a point to note here is that enzymes are important to convert the stored food to absorbable nutrients, such as in the seed, you may have starch that is stored, and the starch will be converted to glucose that can be absorbed by the embryo. We also will have oils and fats be stored in the seed as well, and so those will also be converted into fatty acids that can be used by the embryo. And of course, other nutrients will be there as well. Now for part 3, is a state 2 roles that the moisture from the cotton wool plays in the process of germination. So the moisture, which is water, will do a number of things. One is that they soften the seed coat, which we call a tester. And the purpose for that is to make the seed permeable to oxygen that is going in, and also permeable to the flow of nutrients as well. The moisture from the cotton wool also activates enzymes. And so the enzymes now are able to carry out the chemical reactions, or what we call enzymatic activities. And there are a number of activities taking place in the seeds that require enzymes: the growth process, the conversion of stored food into nutrients, all right, and also other chemical reactions as well, such as respiration. A third point, it also helps dissolve and absorb nutrients, because water will help to transport the nutrients into the embryo and also to dissolve the nutrients before they are absorbed. All right, so moisture plays a lot of important roles within a germinating seed. Now for part B, is that the corn seedlings which germinated are placed in a pot and exposed to unidirectional light as shown in figure 2. And the term unidirectional light, it means that light is only entering the plant from one direction. All right, as the diagram will show you that there's also a slit in this carton box. So light is only coming from this direction only. So that's what it's called, unidirectional, all right? All right, and so part 1 of this question now said, what type of movement is shown by the plants in figure 2? And so it's a tropic response, all right? And to be specific, it is a phototropic response, all right? Or phototropic movement, all right? Because it is responding to light. Now it says state a hypothesis that the student could test by using the apparatus and materials shown in figure 2. And so an hypothesis here could have been to determine the growth response in plants or corn seedlings to light. All right, so they're testing it to light, or to test how light affects the growth of the corn seedlings. So whichever way you state it, you'll be fine. All right. And so for part 3, so the students want the seedlings to grow straight towards the ceiling. All right, it suggests how they could manipulate the apparatus to get this result. So instead of bending, what can they do to make sure that the ceilings go straight up? And they want to place the light directly above the ceilings. So put the light on top of the box rather than the side. All right. You can also cover the tips of the ceilings with an opaque object. That means the auxin will not be moving to no darker side, or it will pretty much distributed only in the tips of the plant because it will be totally covered and dark. All right. All right. So next part of the question, which is part 4, is that the ceilings shown in figure 2 use all the nutrients stored in the cotyledons. Discuss the process by which these plants can make more nutrients. And so I want to point out that the cotyledons, they store food, all right? And so once the food is being used up, then that means the plant now is ready to photosynthesize, all right? So if you notice, the first point is that the ceilings started to photosynthesize, not because they have their plumule developed in the leaves, all right? And of course, as you look at germination, as seeds germinate and start to develop leaves, the cotyledons, they will shrink because they are less active and the nutrients already used up out of those cotyledons. Now, for a second point here, is that water is absorbed from the roots and transported to the leaves through the xylem vessels. So that's, you get water from the soil or from the cotton wool, and then it is transported to the leaves and it goes through the xylem. And next will happen is that carbon dioxide now enter the leaves through the stomata. Light is absorbed by the leaf, and of course light is absorbed by the leaf, specifically the chlorophyll that is present in the chloroplast. So you could add these, that in. These are just points as guide to help us answer the question, so you can actually expound on these points, right? And so the light is needed now to split water molecules, and when water molecules are split, what you get is hydrogen ions and also oxygen. But of course, the oxygen will be given off as waste, but the hydrogen ions, now they will react with carbon dioxide to produce glucose, which is the food or nutrient that the plant needs. And of course, this process again takes place in the chloroplast as a point to note. And again, you can kind of rearrange your points differently and explain it, but you make sure that it's flowing and is consistent with the process of photosynthesis. All right, so for part 5, and I want to point out something as well. Please use the points as your guide in terms of how many important points you need to make. It's not about the amount you write, however, it's about the quality of what you write. So make sure you're precise in your answers, all right? So you don't need to write in no epistle or no extreme long paragraphs and long sentences and so on. No, the points are important. Now for part 5, is to discuss the process by which the raw materials required for making nutrients reach the leaves of the plants. And so I make sure here point out the raw materials needed. And so we need carbon dioxide, we need water, all right? And I'm going to talk about light at the bottom, all right? And so we start about raw materials. Raw materials are generally carbon dioxide and water, all right? Light is an energy or energy source. And so here now, carbon dioxide will diffuse into the leaves through the stomata from the atmosphere. So that's how carbon dioxide reaches into the leaf. And once it's in the leaf, it will go through the air space within the spongy mesophyll layer. So you could add those things in, and then it will diffuse through the leaf and go to the target cells, such as the spongy mesophyll cells and the palisade mesophyll cells. All right. Now, water, however, will enter the roots, and water enter the roots through the root-hair cells by a process called osmosis. And of course, due to transpiration now, or transpiration pull, right? Then, matter of fact, you can even include here capillary action. So due to capillary action, due to transpiration pull, due to evaporation of water, water will create a consistent flow or a continuous flow from the roots into the leaf. All right, and the reason for this, you can also add to this because water is cohesive and adhesive. All right, and so those are very important things you can add in your explanation. All right, so again, it's about writing the correct scientific concept in terms of what is happening to each molecule. And notice I put light here as a point to just explain light. And so light will reach the leaves by radiation and then absorbed by the chlorophyll in the chloroplast. All right, but again, light is just a source of energy needed to split water molecules. So the hydrogen ions are available to react with carbon dioxide. All right, so that's the purpose of light within the leaf. All right, now for part C, is a starch is the most common carbohydrate that is found in the leaves of green plants. Describe a test that the student could do in the laboratory to investigate whether starch is present in the leaves of the seedlings. And this is a general starch test in terms of testing leaf for starch. And I also have a lesson on that if you want to just run over when you're finished and check it out. And so one they have to do is to boil the leaves until the leaf is soft. And of course, that requires a few minutes. Some leaves may go faster than some depending on the leaf, but generally just boil the leaf until it becomes relatively soft. Not too soft, however, not till it cook, but soft enough. All right, and then you're going to remove the leaves now and place them in separate test tubes. Next, you're going to cover the leaves with alcohol and then boil the alcohol until the leaves now are white. And the reason why it becomes white is because the chlorophyll is being removed. All right, so the alcohol will dissolve the chlorophyll and remove the chlorophyll from the leaf. The next thing you want to do, you're going to remove the leaves from the test tube and then rehydrate them, which means they become soft again. And you do that by placing the leaves in the boiling water. Next, you're going to do is to remove the leaves and place the leaf on a mat or on a table, on a napkin, whatever you have in the lab. And then you're going to add a few drops of iodine to each leaf and note the color change. And of course, if the change moved from brown or yellowish brown to blue-black, then starch is present. And so I will encourage you to include that part in your explanation because you want to say, okay, look for this color change, all right? And that color change would indicate the positive test for starch in the leaves. All right, and just to make a note that my points are just to guide you in terms of what you'll write. And again, you just to include these points to ensure that you get maximum marks. Again, you can kind of rearrange them in order as though I have them, but make sure you include them in explanation if you're going to test for starch and things like that. All right, so for part 2, it said, name one plant organ other than leaves where starch may be found. And starch can be found almost all parts of the plant, to be honest with you. But of course, some parts will have more starch than others. And so you may find starch in the roots. You know, some plants roots have more starch than some, such as cassava. You may find starch in tubers, like potato tuber and so on. You may find starch in plant stems, depending on the type of plant. You may find starch in seeds as well. All right, so there are a number of answers you could put here. Pretty much there's no wrong answer once you name part of a plant. Well, except the flower. The flower don't really have starch, right? But other from the flower, yeah, almost all parts of a plant do have some form of starch. All right, now the last question in the last section in this question is that instead of starch, animals store complex carbohydrates such as glycogen. Name 2 organs in an animal's body, which store glycogen. And so let me just point out something real quick, is that plants store starch, but animals, they store glycogen as mentioned in this question. That's something you need to note as well. And so for animals, they store the glycogen, which is the polysaccharide, in the liver or in the muscles, all right? And especially for us, we store a lot of our glycogen in our liver, and so the glycogen will convert back to glucose by the action of glucagon, all right? So when the glucose level drops, then glycogen is converted back into glucose. All right, so let's go into question number 2 now. And for question number 2, and this is figure 3, and this represents a Whittaker 5 kingdom classification of living organism. All right, and so it pretty much asks us to fill in these things. I just gonna start it right here. And so if you notice this, you have eukaryote here, so the opposite on the other side must be prokaryote. All right, and prokaryote will be going into Monera kingdom, which is the example bacteria. On the eukaryotes, we have unicellular, so on the opposite side, we must have multicellular organisms, right? And we have here the protista kingdom. An example of protista will be amoeba, paramecium, and so on. Presence of cell wall, and if there's a presence of cell wall, if it's yes, then we know that it will be autotrophs or heterotrophs. if there's a cell wall. All right. And so if it is autotrophs, then you know it could be plant. And if it is heterotroph, then it'd be like fungi, example yeast. And if there's no cell wall, then it's going to be the kingdom Animalia, example fish, and any other animals, of course. All right, so let's now jump into the question now, because I complete that. You already complete that, so we don't move over. That's 5 marks. So part B now is that each kingdom is further subdivided into taxa, with the lowest rank being the level of the species, defined species. And now what is key to define species is that species is a group of organisms that can interbreed to produce viable offspring. And so if they're not the same species, like example, donkey and horse, if you put them together, get mule, but mule cannot reproduce because a mule didn't come from the same species, so the offspring is not viable to reproduce and to carry on the species. All right, so the species, they must be the same, they must be able to interbreed to produce viable offspring that they themselves can reproduce to produce offspring as well. So in other words, the generation can continue. All right, so that's what a species is. And part 2 of this question is using the human species as an example, explain why physical appearance or morphology is not always useful for identifying organisms belonging to the same species. And so the physical thing is very difficult to determine that, hey, we belong to the same species. Look at the human race, for example. All right. And so human phenotype can vary to a large degree, showing vast difference among the human species. An example of that could be height. Some of us are extremely tall, while some of us are extremely short, right? We can have difference in hair texture as well. Some of us here could be straight, could be curly, could be kinky. So there's so many difference. They can look at people and say we are so different. You know, just barely even eye color, if you think about it, your body size and type. So there are so many difference among the human species. It's trust me, it is. We can describe so many different things. Your finger shape, your shape of your toenails, your eye color, your ears, your nose. There's so many different things on us. Trust me, it's a lot. All right, if you take time to look at the different characteristics, you realize that we are so different phenotypically, but we're the same biologically and chemically because we're made of the same chemicals, right? All right, so part 3 is to suggest one reason why biologists use biological concepts of species instead of physical characteristics to classify organisms. And so to use species instead of physical characteristic, again, why is that some organisms may have similar features but are unable to interbreed. For example, you can have horse and donkey. They have 4 legs. They almost look alike pretty much, but they are unable to interbreed. So you can use the physical features, it's okay, because you have 4 legs, you can interbreed. For human beings and apes, for example, right, or the chimps, we have similar features. We may have mammary glands, we have 2 legs and things like that. You know, we make pretty much upright for the most part, but we can't interbreed. All right? So that's very important because, again, the species now is just more than the physical features, right? And so species is more specific than just physical features, just to mention that. And so different groups of organisms can have similar adaptations but are unable to interbreed. And when we say similar adaptations, we're talking about similar features that allow them to survive in their environment. Even some birds, if you think about it, birds, they look so alike in their features for the most part, but not all birds can interbreed with each other to produce viable offsprings because they belong to different species, right? So it's very important that we use this concept, the biological concept of species, rather than the physical features. Part C is that some lizards were washed away on a log during a hurricane and landed on a remote island. Several years later, they were classified as a new species. It's using the biological concept of species, state 2 reasons why they were reclassified. And there are a number of things we can talk about here. Is 2 points, but just run through these points. One is that they do not interbreed with other species. So that's one reason they're on a new island. And so they are there for years. They're not found in anywhere else, more likely based on what is happening, based on the scenario. So they're not able to interbreed with other species. So yes, they must be classified as a new species. They may develop new adaptations because they're now on a new island. So they may adapt new methods of doing things. And so the way of doing things, even their reproductive cycle, may be different from other lizards. And so therefore they may be classified as a new species. Again, for a third point right here is that they are reproductively isolated from other lizards. And when we say reproductive isolation, we're talking about size, for example. We may talk about the season or the time which they mate. Those are things that can isolate organisms reproductively. For example, dogs. Let's talk about dogs. A pit bull, for example, or a German shepherd, they're not able to reproduce with a chihuahua, right? And simply because the size is different, it's difficult. So they are reproductively isolated. That's what we mean by reproductive isolation. Many things that prevent them from coming together to have a child or offspring. A fourth reason here is that they may have distinct evolutionary changes. And they kind of match up with number 2 if you realize it, because adaptation pretty much intertwined with evolutionary changes. So the changes that they may adapt over the years may not allow them to reproduce and produce viable offspring with other lizards. And so, yes, you have to reclassify them as a new species. All right, so part 2. Is that the new species of lizards living in the mountainous region of the island were found to be smaller than those living on the lowlands. Suggest 2 reasons for this variation. And there are a number of things I can talk about here as well. Now, in the mountainous area versus the lowland, just a few things to point out. Why the ones in the mountainous areas are smaller? One, it could be because of limited food. All right, and so because limited food will cause less growth and development as well, compared to those that on the lower land, they may have access to more food. All right, and this is again is just a suggestion, as they say, suggest the reason why this is possible. We're not saying it is the reason. All right, and the second thing here is that on the mountainous region, you may have more predators. And so since you have more predators, they may not have enough time to grow to full size. All right, compared to the lowlands, they may have less predators and they are allowed to grow to full size or a longer period of time where they can grow in their full capacity. Another reason here it could be that the mountainous lizards, they are more active. And since they're more active, they're going to store less food. So they're not going to be as fat or big. And again, in the mountainous areas, they may have to be hunting a lot and jumping from trees to trees to find food. And so because of that, they are small in size compared to the ones that are less active. They store a lot of food, and so they gain a lot of body fat and so on. So that is now something that can be influenced by the environment. Another one here, which is the last one I think of, is that the allele for smaller size. So the one in the mountainous region have the alleles for small size, while the one in the lowlands have the alleles for a larger size. And this now is a genetic influence. So the first 3, 1, 2, and 3, those are influenced based on the environment. And the fourth one is because of genetics. All right, because if you only have alleles for to be small, it can only be small. If you have alleles to be large, you're going to be large. And so those on the mountainous region, they will keep interbreeding and producing only small size, while those on the lowland, they may have the alleles for large, have to be large, and so they interbreed and only produce large offsprings, right? Or the ability to become large. All right, so those are some of the possible reasons there. Now for question number 3. And this one state now that figure 4 illustrates the event that takes place during the human menstrual cycle. We have 2 hormones here, hormone X, hormone Y. And I know by now you know what these hormones are based on the levels that are displayed on the graph. And here we have the uterine lining. And so I just want to point out something real quick, is that there are 4 hormones that actually work in the menstrual cycle. And we are talking about FSH, which is the follicle stimulating hormone, and we have LH, which is luteinizing hormone. and those are produced by the pituitary gland, the anterior pituitary gland, to be specific, while estrogen and progesterone are produced in the ovaries. All right, and they work in the uterus. So the hormones FSH and LH they're produced by the pituitary, but their effects are in the ovary. While estrogen and progesterone, they are produced in the ovary, but their effects are in the uterus. And so those are small things that you need to know, right? All right, let's jump to the question and say, identify the hormones X and Y and the event occurring at Z. And so X, which is the first one, is higher on the first part, that is estrogen and progesterone, which is Y, higher on the second half of the cycle. And the event Z, notice an egg is coming from the follicle. And so that is ovulation. All right, so those are the three answers there: estrogen, progesterone, ovulation. Now for part two, we said, what is the role of each hormone in the menstrual cycle? And so the roles are pretty much similar, because again, they act upon the walls, which is the endometrium of the uterus. And so estrogen repairs and thickens the wall of the uterus. So its job is to really make it, you start repair, vascularize it, and make it thicker to prepare for implantation. And progesterone now will maintain the thickness of the wall of the uterus, just in case if a fertilized egg is there and implanted, then placenta could be developed and the uterus will be healthy. The wall of the uterus will be healthy enough to support development of the fetus. All right, now for part three, it said name the organs that produce hormone X and Y. Now, the thing is that they are produced by the same organ. They are produced in the ovaries. All right, estrogen and progesterone, they're produced by the ovaries. However, though, I just make a notation right here, is that progesterone is produced specifically by the corpus luteum. All right, and just to make a note of that. But it's still in the ovary, okay? And it said organ here, so that's a very important point as well. All right, so that's why ovaries will be the answer for both. Now for part four, it said discuss the fate of the following structures shown in figure 4 if fertilization of the ovum occurs. So if fertilization occurs, then the corpus luteum, it will remain intact and secrete progesterone. All right, and point to note is that once the placenta develops, the corpus luteum now will start to shrink and disintegrate, because the placenta now will able to produce some amount of progesterone to maintain the pregnancy. All right, or to maintain proper healthy endometrium. Now the uterus lining, which is the endometrium they're talking about here, it becomes thicker to accommodate the implantation of the zygote. So once fertilization takes place, then the endometrium will become thicker, and that is really to accommodate implantation and development of the fetus as well as the development of the placenta. All right. Of course, the opposite is true. If there's no fertilization, then the corpus luteum immediately start to shrink, and the walls of the uterus will start or prepare to disintegrate. All right, so part B is some contraceptive pills contain hormones, suggest how this may prevent pregnancy. And so if hormones are present, what will happen here is that they prevent ovulation from taking place. That's a general concept for contraceptive pills, is really to prevent ovulation from taking place. All right. It also has some other effects as well, such as to increase the mucus built up on the cervix to prevent sperm cells from passing through the cervix up into the fallopian tube. So yes, it does a number of things, but one of the major things is really to prevent ovulation from taking place. Now for part C, it's a drug company is developing a male contraceptive pill. It suggests 3 ways in which this could prevent pregnancy, and they say it's a good thing. So it's what is a possibility or how this may possibly work. And so think about a male taking hormone or hormonal pills to prevent fertilization from occurring. And so one thing could happen here is that the pill could be designed or developed in such a way that it prevents the production of sperm cells, because remember now, you need testosterone to produce sperm cells. So it can inhibit testosterone. So again, it can expound on these points. So the hormones in the pill could inhibit testosterone, preventing sperm production. Also, the chemicals or the hormones present inside of the pills could also work in such a way to reduce the mobility of the sperm cells. Because if the sperm cells can't swim properly or swim backwards, then it can reach up in the fallopian tube to cause fertilization of the egg. Again, point 3 here, which I pretty much can explain. Well, it's different, to be honest, because this one inhibits the enzymes, not the hormone, but inhibits the enzymes in sperms needed to penetrate the egg. Because remember, you know about acrosomes in the tip of, which is the tip of the sperm itself, that contains the enzymes. And once the enzymes are inhibited, then they cannot penetrate the egg cell. So the sperm can reach in the egg cell, so the nuclei could fuse and form the zygote. Again, it can inhibit the gonadotropin releasing hormone. All right, and if you think about the word gonad, so gonadotropin hormone is what will cause the gonads, which is the sperm cell, to release. So pretty much similar to number 1 as seen here, as in number 1. All right, so this hormone is very important to release sperm cells. All right, and they ask you this what? 3 marks, so you have 4 things you could choose from. I can't think about any other possibilities. Now, section B. All right, so we're coming down to winding down to the end. So for section B, it said here now, part 1, question 4, part 1. This is a matter of fact, A1. It says explain how structures in the human eye control the amount of light that enters the eye. And so here, just to make a note, is that the iris, this is very important for you to know and understand as well. The iris controls the amount of light entering the eye by controlling the size of a pupil. The pupil is not a physical structure. It is just a space between the iris. So the movement of the iris, whether it be contracting or relaxing, that is what determines the size of the pupil. All right? And so I separate them into 2 conditions or possibilities here, in terms of bright light and dim light. And so in bright light, the radial muscles of the iris, they will relax, while the circular muscles of the iris, they contract. And because of this, the pupil will constrict. And I need to put a point right here. The pupil constrict, matter of fact, decreasing the amount of light entering the eye. And so let me just quickly put that on here, because, yeah, the pupil constrict, decreasing the amount of light entering the eye. As a matter of fact, you can just write that in, right? So let me just put that real quick. I really don't want to spend too much time, because this is a long paper. Decreasing the amount of light entering the eye. All right? All right. So that's that. All right. So in dim light now, in dim light, the radial muscles of the iris will contract. So it's the opposite of what I have up top. The circular muscle of the iris will relax. The pupil will dilate. And if the pupil dilates, then means no more light can enter the eye. And you can see a little bit clearer. All right? All right. So let's jump into part 2. Now, part 2 state now, Jenny enters a dimly lit room and cannot see the colors of the furniture until the lights are switched on. So the room was dim, she can't see the color, but this is interesting now. It says, explain how Jenny is able to discern objects in the dimly lit room, but cannot see the colors until the lights are switched on. So she cannot see the shape of the furniture, in other words, right? And cannot say, okay, this may be the dresser, this may be the bed, this may be a chair, whatever she have in the room. All right. Now the point to make right here is that what does that mean to Jenny? She's coming from an area where it is brighter. And of course, if it is brighter, the pupil is already constricted. That means little light is going in her eye. So this is very important now, is that Jenny can discern the objects in the room because small amount of light is entering the eye, but not enough to produce colors. So what is important here, because so little light is going into her eye and it's pretty much dim or almost dark, then the part of the part of the retina that is active here are those light sensitive cells we call the rods. So the rods are more active in the dim room, and the rods now detect light only in black and white. So it's night vision, in other words. So you can't see colors when you have little to no light. The rods are active. The cones are now inactive for the most part, right? However, now, when the lights are turned on, now the cones now become more active and are able to detect the colors of the object. Because when more light is going into the eye, the cones become more active. And the cones, just to make a note right here, it said the cones are active in bright light. Point to note. It's a white light is a mixture of colors. That's to point that out as well. So when the light becomes brighter and the good white light is shining from the bulbs and to the furnitures, then the white light contains raw GBIF. Red, orange, yellow, green, blue, indigo, and violet. And so each object will reflect its respective color into the eye. Hence, the colors of the object will be perceived or seen. All right? So that's a nice outline to kind of tell you exactly what's happening, where she's coming from, what happened, and so on. That's 5 marks, nice and easy. Now for part B, for this question, it said a certain form of color blindness is inherited as a recessive allele carried on the X chromosome. So this is going to be key. So the allele responsible for color blindness is on the X chromosome. All right, and it said no, it is just said to be X-linked or sex-linked. Now, a woman who is with normal color vision, whose father is colorblind, so this is now very important. So the woman who was normal, is her father is colorblind. And for the man to be colorblind, remember that man only has 2 chromosomes, right? X and Y, sex chromosome, X and Y. And it's carried on the X chromosome. So if the woman's father is colorblind, it therefore means that his X chromosome must have the recessive allele. So let's just put something here to show what I'm talking about. This is the woman's father. The X chromosome will have the lowercase b, right? And the Y, of course, you can just write the Y, it doesn't really matter. And so this will be her father. All right, so let's go back through the question to finish up the question and show you something here now. Now, she mates with a colorblind man, right? What is the chance of them having a colorblind children, or colorblind children? Use genetic diagrams to explain the answer. And I have 2 diagrams to show you 2 different versions of the same thing. Now, if this is the father of the woman, and this is where my concern is, this is the father of the woman. All females gain this X chromosome, the sole chromosome, the sole X chromosome from their father. They must get it. Because if a Y chromosome is present, it cannot be a girl. It's a boy. So therefore means now she get a normal chromosome from her mother. That means her mother could be heterozygous. This woman mother could be heterozygous normal or homozygous normal, doesn't matter. The fact that she gets this night blindness or color blindness allele from her father, but she's normal. That means now our other X chromosome is normal and is dominant. She get that from her mother. All right. So this will be her mother here, and I'm a motherfucker. I'm put one in question because it doesn't matter what this is, but this one that she's going to receive must be a normal. And so the woman in this question must be heterozygous, and that's why she is normal. See it right here? She has normal color vision. And the only way she's going to be color vision if her father has color blindness is because she get a dominant X chromosome from her mother. And she must get the recessive one from her father who is colorblind. So she must be heterozygous. Now, however, if the man is colorblind, just like her father, see right here, the lower case B must be on her husband, X chromosome. So we know now that the woman is this or this. So whichever one you want to write. I'm giving you 2 options, right? The man now has to be homozygous recessive because he is colorblind. The woman is only a carrier. Okay? So a way to do this, and I give you two options here. You can either do it like this or do it like this. So you can put the X and Y chromosomes to represent the male and female. So I put the woman on top of the box, or possible gametes on top of the box, the man's possible gametes on the side of the box. And so in the box, you get these results. You can pause, go through it, make sure you understand it. And if you do have questions, talk to me in the comment section. I'll respond to you as soon as I possibly can. Now, based on the result here, we have 50% chance of being colorblind because the only person could be colorblind. A matter of fact, I label them. They must have two lowercase p's, which means the two recessive alleles. In this same thing is the same exact thing. This is colorblind, two lowercase b's. The lowercase b must be on the X. The Y has nothing because the Y is not carrying the disease or trait. Is an X link, right? Not a Y link. All right. And so for any of them, if you draw this one, the only thing you can say is 50% chance being colorblind because that's what the question asks you to ask you. However, if you draw it this way, you can get more information from this. Yes, 50% of the total offspring will be colorblind, but out of that 50% chance, 25% chance may be a boy and colorblind, which is this bottom one right here that I'm going to tick right now, a boy and colorblind, or 25% chance being a girl and colorblind, which is that one. All right? All right, now let's kind of go down to the next part of the question now. That's a nice question, I must say that. Now, question number 5 is to describe the chemical digestion of carbohydrates, fats, and proteins from the mouth until they are absorbed in the small intestine. And I want to understand something here. Even though they say from the mouth, they use the word here, chemical digestion. All right, let me erase that. All right, so this chemical digestion they're asking you for right here, chemical digestion. And if they ask you for chemical digestion, it doesn't matter where you start, if it's the mouth or not. If chemical digestion is not taking place there, you can't talk about it. Now, I put carbohydrates. And for carbohydrates, you can have different types of carbohydrates, right? And so you can take in sucrose, which is table sugar, right, or normal cane sugar. But if you eat in starch, I choose starch as my carbohydrate. So if you choose starch, then starch will start to break down chemically in the mouth by salivary amylase. And notice I put the mouth right here. It breaks down into maltose, and then maltose will break down by maltase to glucose, and this takes place in the small intestine. All right. Now for fats, and just to make a mention right here too that in the small intestine we have pancreatic amylase that are produced and will work inside of the small intestine to break down any excess starch that actually reach into the small intestine, right? So if you're eating starchy food and all of it is not broken down in the mouth by salivary amylase, or by the time it reaches the small intestine, then the pancreas will produce more amylase that will work on the excess starch to break them down into maltose, and then the maltase will break down the maltose into glucose, all right? So that's the purpose for pancreatic amylase. Now fats. Chemically, fats are acted upon by bile, and bile is a salt, not an enzyme, just to point it out. And this takes place in small intestine. So fat only chemically break down in small intestine, right? Mechanically in the mouth, yes, but chemically, small intestine. And so fats break down the fatty droplets, and then the fatty droplets now act upon by the enzyme lipase in the small intestine again, the fatty acids and glycerol. So glycerol was supposed to be here, right? I think piece of it drop off, but don't worry about it. It's glycerol. Matter of fact, I could slide over a little bit, and you will see it is glycerol, right? And so proteins now, proteins start to break down chemically in the mouth. Sorry, not in the mouth, mechanically in the mouth, but chemically in the stomach. So proteins start to break down chemically in the stomach by the enzyme pepsin. It's a protease, right? And then now break down the polypeptides and also peptide as well. And then trypsin from the pancreas will work on the polypeptides in the small intestine to break them down into amino acids. And amino acids can be absorbed. All right, so the last thing on each of the reaction will be the end products, and those can be absorbed into the bloodstream or different parts of the body. Of course, for fats are absorbed by the lacteal in the villi, not into the bloodstream, just to make a note of that. And so fatty acids will travel into the lymphatic system, not in the circulatory system or in the blood. All right. Now for part B, Here said describe how the body system work together to assimilate glucose molecules. And so there are a number of things. A matter of fact, let me just point out something here, right? And again, I just want to give you the points so you can actually expound on points and kind of guide you. And so the body systems that are involved is the digestive system, circulatory system, lymphatic system, and the respiratory system, right? So 4 systems are involved in the assimilation of glucose molecules. Now, watch this now carefully. As I go through, I think I'm going to kind of relate the system as I go through, right? But a point to note, the very first point to note, I want you to know this, that assimilation means the process by which absorbed nutrients are used by the body or converted to body materials. So either be used or converted to different things in the body. Such as when we eat proteins, protein break down to amino acids, and then we use amino acids to make our proteins. Yeah, that is assimilation. All right, so here now for glucose, assimilation of glucose, this is what takes place. In the digestive system, and the digestive system now breaks down complex carbohydrates into glucose. That's first happen, right? It must break down the glucose first. Whatever carbohydrates you're eating, whether it be disaccharide, polysaccharide, it must break down the glucose. Now the villi, which is still part of the digestive system, will now absorb the glucose molecules in the bloodstream. The bloodstream now, by the circulatory system now, will transport the glucose to the target cells. Now the glucose now molecules are sent to the cells in the body, which is our target cells. And then glucose now start to metabolize in the cytoplasm to form what they call pyruvate. That's the first part of the respiration process. So the lymphatic system, doesn't make a mention here, and I didn't put it here though, but the role of the lymphatic system in all of this is to take the glucose from the bloodstream, which is the circulatory system, into the cell by tissue fluid and all of that, right? It passes through this tissue fluid into the cells. So I just want to make a note of that. That's why the lymphatic system comes in play. However, now the first thing, glucose breakdown in the pyruvate, or I can say simpler molecules, if you want to put that, if you don't remember pyruvate, then the pyruvate now will convert to ATP, which is adenosine triphosphate. And adenosine triphosphate is a usable form of energy that the body can use to make its own energy or to carry out functions for different activities. Now, point to note is that pyruvates are converted to ATP in the mitochondria. All right. And once it's converted to ATP in the mitochondria, then the cells can use them. So here I said, and they are made available to the cells to carry out functions. Whatever the functions the cell need to carry out, yeah, the energy is there, ATP is there. All right. Now for part 2 is to explain how the assimilation of glucose by a non-diabetic person differs from that of a person suffering from diabetes. And so to make life easier, just kind of put them into separate thing. I think this was a 5-mark question. Let me just slide down to it. Yeah, 5 marks indeed. And it's the last part of this question here. And so these are just points to discuss. I just make a note of this. Points to discuss. And so one is that non-diabetic will be faster conversion or faster reaction, which is mean faster assimilation. A diabetic person will be slower conversion of the glucose or use of the glucose or slow reaction of the glucose. And simply because for non-diabetic, you have a higher concentration of insulin. Therefore, that means the reaction will be faster because higher concentration of reactants or enzymes will speed up the chemical reaction itself. Lower concentration will slower the reaction. So if the catalyst is lower, then yes, you have a slower reaction. A point to note here as well is point 2 is that more glucose can be converted into energy or usable energy. Less glucose will be converted to usable energy in the diabetic person. More glucose is taken into the cells. You can even put this before number 2. So if more is taken in the cell, then less will be converted. More will be converted, I should say. So more glucose is taken into the cells for a non-diabetic person. For a diabetic person, less glucose is taken into the cell because insulin is needed to passage glucose into the cell as well. All right? And so in the non-diabetic person, glucose, you have more glucose is able to convert to glycogen, which means you have more storage of the glucose molecules. And so in a diabetic person, you have less glycogen will be formed, and so therefore less storage in the liver or glycogen in the liver or also in the muscles. And of course, that's why persons who are diabetic tend to be tired easily because of a lack of available glucose in the cells to convert to energy, or low conversion into energy. Again, that also ties into the point I just made, that more ATP will be formed in terms of energy availability. In a diabetic person, you have less ATP formed. That means less energy available. That's why they are tired easily. And again, for a non-diabetic person, no glucose will be found in waste, such as in urine. All right. But for a diabetic person now, you have glucose will be removed from the body as waste, and therefore you find glucose in the urine for a diabetic person. So this is how a non-diabetic person uses glucose, or let glucose become a part of the functional body, while a diabetic person now, this is how they actually use glucose, or glucose become part of the body as a usable substance. All right, and again, you just need to remember what assimilation is. Now, our final question, and we are almost there. Now, is a using suitable example of each distinguish between predator-prey relationship and parasitism. Now, here, predator-prey versus parasitism, very important here. Just an example I'm going to use is bird and grasshopper. Bird is a predator, grasshopper is a prey. An example here would be flea and dog. Flea is the parasite and dog is the host. And so, first one here is that the predator will hunt and kill the prey, while the parasite will invade and also live in or on. All right, I say invade, which means go in, or lives on the host. Okay. The predator will digest the prey to absorb nutrients. However, now the parasite absorb nutrients from the host blood or from the host cells. I'm going to slide over a little bit so you can see that, right? All right, so pretty much if you think about it, parasites are lazy. You know, they don't do much for themselves, just absorb, sit and just wait till the host break down the stuff and have it in the bloodstream, just absorb it and just live. That's a lazy attitude. Now, the next one here is that the prey is immediately killed. However, the host may not die immediately, but may die over a period of time. You know, due to complication or sickness or lack of nutrients, they start to show weakness in certain things, and then the organs may start to succumb to the lack of consistent nutrients. The next point to make here is that the predator is generally larger. So predator is generally larger than the prey, generally larger. Of course, you have some, you may have some predator that are smaller than the prey. If you think about a lion, for example, and a giraffe or a deer, you know, sometimes those organisms are larger than the predator. But generally speaking, though, the predator is generally larger, and parasites is pretty much always smaller than the host, right? All right. And so a feeding, our next point here is that each feeding involves a different prey. All right. So every time that the predator needs something to eat, it will be a different prey, because they have to kill, eat that one, and continue to go for a different one. However, now the parasites will feed on the same organism over and over and over and over again. All right. And pretty much, as I said, they live in. If you look at number one, they live in or invades or live on. They don't really move. And so here now, for the predator-prey relationship, there's no reproductive relationship. What do I mean is that it has nothing to do with their reproduction. Nothing to host or enhance. Well, it could be enhanced because the nutrients will enhance reproduction. But in terms of direct effect on the reproductive system, none. However, for parasites carry their whole life cycle on the host. So they lay their eggs there. If they're egg-laying parasite, they have the eggs there, they hatch and all of that. If it's intestinal worms, then they're going to lay eggs or lay eggs inside the intestine and things like that. So they pretty much carry all their life cycle. And so over a period of time, you have more parasites on that host because of the reproductive cycle taking place on or in that host. All right, so that's all the differences between the relationship between predator-prey and the parasites or parasitism. And that's a 6-mark question there. All right, and so we are on part B and we're almost there. Now, it says some environmentalists encourage the practice of introducing predators to an ecosystem to control a population of pests rather than the use of agricultural chemicals. is to outline 3 advantages and 3 disadvantages of this method of biological control compares to the use of chemicals. And so I'm giving you the advantages of the biological control. And so biological control, and again, if you could put them in opposite, if you want to do like that, the advantages versus disadvantages. And so the advantages of biological control, there's no pollution. Of course, if you use chemicals, there will be pollution because chemicals will be, chemical remains or residue will left in the environment. For biological control, there is no bioaccumulation. What do I mean like that? No poisonous substance will remain inside of the organism that will travel on through the food chain or the food web. No actions. Action is continuous, which means if you have a predator-prey relationship, like another organism eating another organism to control them, then it'll be a continuous feeding relationship. You'd have to be applying chemicals, like chemicals, you have to be applying chemicals over and over or in certain time period. But once the predator is there to destroy, or the mechanism, the biological mechanism is there, or agent is there, then they will always be there ready to destroy the pest. All right, and again, another point here, which is number 2, which I kind of go down the list. Another point here is that there is no resistance developed easily because, again, if it's biological, then it's a natural feeding relationship or attack based on if you use bacteria. Because bacteria could be used to control certain insects as well. Usually kill specific pests. So a biological, because based on feeding relationship, all right, or symbiosis, then it will kill the specific pest and not just everything. If you use chemicals and you want to kill, for example, caterpillars, they may kill other useful insects. But for biological control, is very for the most part, it is very specific, right? Now, in terms of disadvantages, the organism that they use or the biological agent can eventually become invasive. A matter of fact, this tie up into number 5, which I'm going to discuss. Again, they may feed on other organisms sometimes, which is rare, but that's also a possibility. They can introduce diseases. Since it's a living organism, they can sometimes carry diseases that you don't know that they have. Again, a disadvantage requires time to be effective because introducing organism or living organism to control another living organism, they may not immediately kill everything. You know, as one of the advantages, that is a continuous thing. All right. And so it may take time for it to be really effective. It may not be immediately. Like today, you spray chemicals now, it may have immediate effect to kill all the bugs or all the pests. Number 5 now may become a pest itself. So what they use to control one pest, it may become so large in number or populating so fast degree that they themselves may become pests for another crop or another animal. If you're an animal, and so they themselves can become pests or become invasive. So they're pretty much similar, but they're kind of different. All right, so invasive differently from being a pest itself, right? Slightly different. All right, and so we have, I think, one other piece more to talk about, and we finish. Yeah, I think we have one small piece to talk about. Right, and so here we go. Is explain how biological control may contribute to biological evolution through natural selection in an ecosystem. And so let's just run through these points real quick and we out of here pretty much. And so the first point here is that for natural selection to occur, the prey must display variation. There must be variation for you to have natural selection. If everything is the same, then you can't have natural selection. You cannot. And so the variation could be such as in color, size, speed, or even behavior. Now the second point to note here is that the predator might only be able to see or catch a specific prey. For example, if one of the organism or variation of the organism may have a color that camouflage them, then they're going to be difficult to be killed by the predator, right? And so next point here to make now, if the predator constantly feeds on that particular prey, that is one that they can see or catch, then that variation of the prey may become extinct. And next point to note here now is that the surviving variation of the prey will reproduce, giving rise to the existence of that species. So over time, you're only going to see that particular species in that habitat or ecosystem or environment. And so we're at the end of it. I really thank you for being here. Please take good care of yourself. Be good on the exams. I wish you good luck as well. And please, if you haven't at this point, subscribe. Also share with your friends. And I will talk to you soon. Take care. All right. See you again.